Alright, this video is going to talk about DNA replication. I'm going to start off by drawing my parent strand and giving it some directionality, so making sure that it's anti-parallel. Remember, the 5 and the 3 are just referring to the very last nucleotide in the nucleic acid strand. A nucleotide is composed of a sugar, a nitrogen-containing base, which is going to be either 1 ring or 2 rings, depending on whether it's a pyrimidine or a purine, and then finally our negatively charged phosphate functional group. So our carbon atoms are 1, 2, 3, 4, and 5. And so that's what these numbers 3' and 5' are referring to. So what I'm going to do in order to initiate replication is I'm going to break the hydrogen bonds that are holding these 2 strands together. The hydrogen bonds are going to be between the adenine and thymine, and guanine and cytosine. And I am going to create a replication bubble, and we're going to call this the replication bubble. This is going to be the origin of replication, where I'm going to start laying down some primers. So the primers, let me get my directions down. The primers are short segments of RNA nucleotides. Remember, RNA nucleotides at that second carbon have a hydroxyl. If we were talking about DNA, they just have a hydrogen at that second carbon. A primer is laid down by the enzyme primase. Another enzyme that you need to know that I didn't include is helicase. Helicase is the enzyme that's actually going to be breaking the hydrogen bonds and opening up the nucleic acid strand. And there are also proteins called SSB, or single strand binding proteins, and that's going to keep these strands from coming back together. If these strands come back together, then we haven't made a copy. So I'm going to lay down the primer by primase, and the primer is always going to start as a 5, and it has to move from the 5 to the 3. So we always, this is a huge rule, we always, when we're synthesizing new DNA or RNA, we are always synthesizing in the 5' to 3' direction. The reason for this is that the 3' or the 3 carbon has a hydroxyl, so that is where the dehydration reaction is going to happen. So this 3' with this OH sticking out, when we add another nucleotide to it, we've got the 3' sticking out. If I were to add another nucleotide to it by a dehydration reaction, I would have a 3' OH sticking out. So we're always going to be elongating from the 5 to 3. So in order to make this primer anti-parallel, I would not go this way. Going this way produces a strand that is parallel to the parent strand. That's no good. So I've got to go to the right on this top strand, and on the bottom strand I've got to go to the left. That gives me an anti-parallel relationship between the daughter strand, which is going to be in red and blue, and the parent strand, which is going to be in black, or which is in black. Okay, so I've got my hydroxyl poking out of the 3' end. Now DNA polymerase is going to come in, and it's going to work off of that primer and add new DNA nucleotides in a complementary fashion. So in other words, where if this were an A on the parent strand, it would lay down a T. If the next nucleotide on the parent strand were a G, it would lay down a C, and so on and so forth. Off it goes, laying down new nucleotides, synthesizing that daughter strand until eventually it just runs off of the DNA molecule in this figure. Now in reality, it's just going to keep on going until it runs into another replication bubble. We call these the leading strand. And the reason for that is because this strand is synthesized continuously. There is no breakage. That is not true for the lagging strand. So the lagging strand, here's the problem, is that we have to synthesize DNA in a 5-to-3 direction, which means we can't go this way. Because if we go this way, then this portion of the daughter strand, this entire portion of the daughter strand on this side and on this side are going to be parallel if we just extended the primer out in this direction. And parallel DNA is no good. So we've got to synthesize in a 5-to-3 direction, and the only way that we can do that is we have to wait for this fork, this replication fork right here, to elongate or to extend far enough so that there's just room to lay down a primer. So we'll lay down a primer here. That's the 5 end, and we'll go this way. That makes up the 3 end. And now we have enough room for DNA polymerase to come in and start adding nucleotides in the 5' to 3' direction. So this enzyme is called DNA polymerase 3. And what's going to happen when it hits this primer that was laid down previously is it's going to fall off. It's going to fall off. And our new 3' end is just this nucleotide right here that has an OH sticking out. And we call this fragment that is produced an Okazaki fragment. A different enzyme called DNA polymerase 1 is going to sort of latch on to that 3' end, and it's going to go over the primer and it's going to erase it. It's going to replace the RNA primer with just DNA nucleotides. And that's done by DNA polymerase 1. And then the final enzyme that's emphasized in the DNA replication process is there is a break in the sugar phosphate backbone between these two nucleotides. And an enzyme called ligase is going to come in and it's going to do a dehydration reaction between those two nucleotides in order to seal the sugar phosphate backbone. And what we end up with is one long continuous strip of daughter DNA. And so you're seeing that, let me see this real quick. That's what you're seeing in this figure. You're seeing a 5-to-3 primer. DNA polymerase is latching on to that 3' end, and it's going to travel along synthesizing DNA until it hits this primer that came before it, and it's going to fall off. DNA polymerase 1 is going to erase that primer and replace it with DNA nucleotides. And then finally, ligase is going to come in and staple by basically doing a dehydration reaction and seal this sugar phosphate backbone. And so that's how it works. Let's look at how it would work on this top strand. So the replication bubble has opened enough so that we can lay down our 5-to-3 primer. DNA polymerase 3 docks on, begins synthesizing, falls off. This is our new 3' end. I guess I should draw that arrow a little better. This is our new 3' end. DNA polymerase 1 is going to latch onto that 3' end and begin erasing the primer and replacing it with DNA nucleotides. And then finally, ligase is going to create a covalent bond between this nucleotide and this nucleotide. And that's how it works. So what's pretty interesting is that when we get to the end, here's our 5 to 3 primer. I'll do it again. DNA polymerase 3 latches on, falls off when it hits the next primer. DNA polymerase 1 comes along and replaces the RNA with DNA, falls off right there. Ligase creates a covalent bond to seal the gap. If you look at the very end of the DNA, this is the 5' end, right? Get rid of all these. Our 3' end is all the way over here. This will be our 3' end. What's interesting about this end of the lagging strand is that DNA polymerase 1 can't erase this. Remember, the only way that DNA polymerase 1 is able to erase the primer is if it can latch on. Let me see if there's a better picture that talks about if it can latch on to this 3' end. Hopefully you can see my cursor in this video. So we need to latch onto this 3' end, and when it does that, it can erase the primer, which is exactly what it did right here. But notice that there's no 3' end to attach to of an Okazaki fragment. There is no 3' end to attach to erase this primer. And so what ends up happening is this primer breaks down and you end up with just a single strand of DNA. And that's no good. The cell does not like single-stranded DNA. It indicates that something is wrong, and it could result in apoptosis if this single strand survives. So what the cell needs to do is it just needs to get rid of it. It needs to get rid of that single-stranded DNA, and the same is going to happen over here for this lagging strand. We're going to see primer, polymerase 3, fall off, right? This right here is a 3' end of an Okazaki fragment, which means that polymerase 1 can latch onto it and erase this primer. Ligase comes in, seals the backbone. I know I'm repeating myself, but so there's no 3' Okazaki fragment, which means we need to get rid of that primer, and now we also need to get rid of that single strand DNA that's hanging out over. Okay, so what this means is that every time the DNA is copied, it gets shorter. So you would think that if we're looking at just a long double-stranded DNA, this is a long double-stranded DNA, if every time the DNA gets shorter and shorter and shorter whenever the DNA is copied, then isn't the cell cutting away precious genetic information? And there's a solution for this, and the solution is at the end of a DNA strand, the cell has a sequence of nucleotides called telomeres. And these telomeres are nucleotide sequences of, I believe, TAG, or maybe TAGG, or something like that. But it's TAG over and over and over and over again. And so that way, and this isn't coding for any protein. That way, whenever the cell divides, it loses a little bit of its telomere. But that's okay, because it's a buffer. We're not cutting into any precious genetic information. And the cell has enzymes that enable it to make new or more telomere. Those enzymes are called telomerase. But what's interesting is that a lot of cells don't make new telomeres. They aren't producing telomerase. And so after cells divide for X number of times,
they enter a state called senescence, where they will stop dividing. That's called there's a word for it, and it's called the Hayflick limit. It's the number of times a cell will divide before undergoing senescence. And we'll talk about some cool applications for that another time.